Okay. Happy Friday. We made it to Friday. I counted, we have 8 classes left. Not that you're counting, but I'm counting. There are 8 classes left. One of them being today, one of them will be a test, which means we have 7 classes left to cover 3 chapters. I feel okay about that. 7 classes to cover 3 chapters. Your chapter 16 homework is due on Monday. That's on Moodle. There are some links on these slides that I think will help you with the homework. But we're talking about a little bit more in detail than maybe I think I originally thought we would, so you might not need those as much. I don't know. Questions about schedule stuff? Okay, then we are going to jump right into chapter 16, which is the molecular basis of inheritance. So we talked about genes and alleles, we talked about chromosomes, and now we're going to talk about inheritance at a DNA level. So we're going to wrap up the semester with chapter 16, 17, and 18, which are covering the central dogma of biology, which we talked about a while ago, but let's see what you remember. The central dogma of biology says DNA makes RNA, RNA makes proteins. So this picture looks better on my screen. This is showing you the central dogma of biology. DNA, which you can't read, makes RNA. So this is our 3 chapters that are coming up. So DNA also makes DNA. So that's chapter 16. We're going to talk about the process of how DNA makes more DNA. DNA makes RNA. This is the process of transcription. Because we're taking DNA language and we're putting it into a different version of the same language. We're still in the language of nucleic acids. We're transcribing DNA to RNA. So that's chapter 17. And then the last chapter that we will cover together is chapter 18, which is translation, where we take that language of nucleic acids and we translate it into the language of proteins. So this is the summary. This diagram summarizes the rest of the semester for us. DNA makes DNA, but DNA also makes RNA, and RNA makes protein. And that talking thing is right over. Sorry. So we're actually going to start out chapter 16 with some history. About how did we figure all of this out? The point of this is, one, to show you that we figured it all out scientifically, but also just to think about the scientific process. How do we have a question? How do we approach that question? And how do we use science to help answer that question? And to show you that our understanding of things changes over time. Science changes its mind, and that's okay. So if you printed the PowerPoint, sorry, I added a bunch of pictures in later, like at 5:30 this morning. And I didn't post them, so. We're going to talk about some of the different experiments that were used to figure out, number one, that DNA was a hereditary molecule. We didn't always know this. Okay, you have known this your whole life. I have known it my whole life. I mean, maybe not like from birth, but it's been known my whole life. But it wasn't always known. It's only in the last 100 years that we figured out, 1, DNA is a hereditary information. 2, what does DNA look like? And 3, how does DNA become RNA and proteins in that process? So we're going to talk about a series of experiments that helped us to figure out what DNA looks like and what DNA does. So the first couple experiments are talking about how did we know that DNA is the hereditary information. So at the point when Griffith did his experiments in the 1920s, his main experiment was in 1928. At that point, there was still discussion there was an understanding of heredity because Mendel had already made his discoveries and Morgan was making discoveries, and people understood heredity as a thing. They understood that traits were passed on from one generation to another, but they didn't know why. And so they didn't know what molecule made that happen. They didn't know what part of the cell made it happen. They knew DNA existed, They knew other things existed, but they didn't know that it was DNA that was carrying that information on to the next generation. And so the question at this point was really, is it proteins or is it DNA? So they kind of were down to proteins and DNA. And honestly, a lot of people, a lot of scientists at that point were leaning towards proteins as the genetic information, as the molecule that was carrying genetic information to the next generation. Why do you think they might be leaning towards proteins? It's Friday, I'm asking them. Okay, how many, the structure of DNA was known a little bit. So how many bases are there in DNA? 4. Okay, there's 4 bases in DNA. It doesn't seem to provide enough material for genetic diversity as what we see in the living world. From bacteria all the way up through humans, we're talking about 4 bases creating that diversity. On the other hand, proteins have 20 amino acids that they're working with. There's 20 different amino acids that are parts of proteins, and there's lots of different sizes of proteins and lots of different forms of proteins. And so a lot of the scientists are leaning towards proteins as this must be it, because look at how many proteins there are and look how different bacteria are from people. And so Griffin and a bunch of other people were trying to prove or figure out this, but most people at this point, a lot of people were leaning towards proteins rather than DNA. So Griffin was actually not trying to figure this out. Griffin was trying to develop a vaccine for Streptococcus pneumoniae, which causes pneumonia. And so you probably have some knowledge of vaccines, especially earlier vaccines, where the idea was you would take something like a virus and put it into a killed virus and put it into the body, and the body would conjure up some kind of response and then have a memory of that, so that if you came into that contact with that virus later, your body would say, hey, I know what this is, and I can attack it and get rid of it. So he was working with different strains of the Streptococcus pneumoniae bacteria, trying to make a vaccine. And so he found out there are 2 strains, the R strain and the S strain. So the R strain was called the rough strain, and the S strain was called the smooth strain. And the rough strain had no capsule on the outside. And the S strain had a capsule. So only one of them is what we call pathogenic, and that's the S strain. What do we mean by pathogenic? Causes disease. So there's 2 forms of this bacteria, 2 different forms of bacteria. Only one of them is pathogenic, meaning it's causing pneumonia, which is causing a problem. And it's because of that capsule. That capsule makes it resistant to the immune system. So the immune system can't fight it like it can one without a capsule. So he tried different variations, again, because he's trying to come up with a vaccine. He's working with mice. And so he injected some of them with the rough strain. And the rough strain mice were injected with these bacteria, and they lived and they were fine. He injected some of them with the smooth strain, which they got pneumonia and they died. So that to him was that figured out this. There were 2 different strains. Some are virulent or pathogenic and some are not. So then he tried heat killing the smooth strain, because again, that's what you want to put into someone for a vaccine. You usually use a dead virus. And so he injected that and the mouse lived because there was nothing, the bacteria were killed enough. The mice did not have a reaction to it. It didn't cause an infection. But then when he took this rough strain and he mixed it with some heat-killed smooth strain and put those both into the mice got sick and the mice died. So putting both the rough strain and the dead or heat-attenuated smooth strain, putting them together into the mouse caused the mouse to die. Why do you think that happened? Guess what? He didn't know. And you may not know either. He figured out that there was something going on that he called transformation. That somehow, even though those viruses had been treated with heat, they were able to pass on information to the live rough strain bacteria. And what he found was in the mouse, in this dead mouse, he found live smooth strain bacteria. So even though he only put in the heat killed bacteria, he killed the bacteria. He found live virulent bacteria, even though he hadn't put any live ones in. That makes sense? So he figured that somehow those dead bacteria were able to pass on information to the rough bacteria that made them into smooth bacteria, so that then they died. The mice were affected and caused infection.
The heat kills smooth.
It's it's bacteria that have the capsule, but he killed them. So when you get a vaccine for an illness, most vaccines, not all, are like dead. They put in pieces of like the cell membrane or the cell wall that causes your body to have a reaction. So your body still recognizes that this is not good, this isn't something that's supposed to be here, and causes a reaction in your body to remember it. So he's killing the S bacteria. And when they're totally dead, and that's all he puts in the mice don't die because they're able to fight off the infection that's not really an infection. But when he mixes live rough strain with dead smooth strain, the mice get infected and then end up dying with live bacteria in them. So he figured out that those bacteria that are dead are somehow able to pass on information to the rough strain one. It's genetic information because the next generation of bacteria is S bacteria. Jamie, did you have a question?
I was gonna ask, so how did the dead strain pass on the genetic information to the live strain who's dead?
So we're not gonna get into all the technicalities of that, but there can be like plasmid DNA that can be released when the cell dies. That can be taken up by other bacteria. So bacteria have the ability to take DNA out of the environment and then transcribe and translate it into proteins. So it's able to take the bacteria out of the dead bacteria, like if the cells are lysed, it can take pieces of that DNA and express it. Okay. Jeff, in bio 2, we talked a little bit more about that. So from this, the basic gist of this was that he was able to figure out that this transformation was somehow inherited. And so that bacteria was passing on information to the next generation. Even though it wasn't even alive, it was able to pass on some kind of inherited pathogenicity, if you want to say that word. That makes you sound like you know what you're talking about. Inherited pathogenicity. So from that, further experiments happened. Avery, McCarty, and McLeod continued these experiments. And basically they were taking extracts of the serums from the mice. So they took serums from the mice and they added them enzymes to them because they were trying to figure out what was it about the cells that they were passing on. So to some of the serums that they took from the mice, so this is mouse serum. So they have these heat-killed S cells. So those are the ones that he mixed with the R cells to put in the mice. So they added proteinase. So ACE tells us it's what? It's an enzyme. It's proteinase. So it's going to break down protein. So they added this enzyme, proteinase, that breaks down the protein. When they added the R cells to the S cells, they still got live S cells. So proteins are broken down. They still saw transformation. Okay, they also added to another tube, they added RNase, which would tell you ACE, it's an enzyme, RNA, ACE breaks down RNA. So these have no RNA. Mixing the R cells with that, still got the S cells. And the third ones, they added DNase, which is an enzyme that breaks down DNA. When they break down the DNA and they add the R cells to the S cells, they didn't get any more S cells. So by removing the DNA, the transformation didn't happen. So when DNase happened, there was no transformation. Wow, I can't spell today. So when they used an enzyme that took away the DNA, there was no transformation. When they took away the proteins, there was still transformation. When they took away the RNA, there was still transformation. So that further proved that this was DNA that was passing on the information to the other cells. So DNA is the molecule that's carrying the heritable information on to the next generation from the S cells to the R cells. Does that make sense? So that was their experiment was 1944. So that was 16 years later. And then we get to Hershey and Chase, which is possibly one of my favorite experiments ever because it involves a blender. Okay, science, we think of high-tech machinery and stuff like that. They just used a blender. Okay, they had other high-tech stuff. I don't want to say they weren't doing high-tech things, but yes, this is the picture from your book that is taken from Quizlet. So someone blacked out the words to try to quiz themselves on it. Okay, so Hershey and Chase, we're working with bacteriophages. Again, still trying to prove that it's DNA that is the information that's passing on the information. So bacteriophages are viruses that affect bacteria. Bacteria, wow. Affect bacteria. So bacteria can get sick too. They can be infected by viruses, just like you. So they had phages. They worked with a specific phage that affected E. coli. E. coli is a bacteria that lives in your intestines happily most of the time, sometimes not so much. If you've ever had food poisoning, you can have E. coli food poisoning. There are certain strains that are pathogenic. But most of the time, you and your E. coli and bacteria and your other bacteria in your intestines are happy. You work together. It's a good relationship. So they took the T2 phage. So bacteriophages, we call them phages for short, because they have a nickname. So bacteriophages, T2 bacteriophage affects E. coli. So they did 2 different batches of this experiment. So they took phages that were grown with radioactive, sulfurs, and some that were grown with radioactive nitrogens. And now I'm doubting myself that I'm remembering which one is sulfur and which one's nitrogen. I think I'm 99% sure I'm right on this. Okay, so phages, they know phages are made up of 2 things. They have a protein coat and they have some kind of nucleic acid, whether it's DNA or RNA. So those are the 2 ingredients of a phage. So they were trying to see, again, is it the DNA that's carrying on information or is it the protein? Because viruses are only made of protein and DNA, it was a relatively easy experiment in terms of thought process. So they labeled some of the protein coats with radioactive proteins, with the radioactive sulfur so they could track it. And they labeled the DNA, because T2 is a DNA phage. They labeled the DNA with radioactive nitrogen so they could track it. I am right. I did have it in my notes. I didn't see it. It is sulfur and nitrogen. So they then introduced these phages to the bacteria. So these bacteria have radioactive proteins. And these bacteria have radioactive DNA. So they let the phages infect the E. coli. And then they stuck them in a blender. After a while, they blended them up, and that blending separates the DNA. The phages from the E. coli. So now they have phages and DNA floating around, phages and bacteria floating around in this liquid, and then they centrifuge it. So they spin it down really, 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 really fast. And this is my goal by the end of the semester, is to stop accidentally advancing slides. Okay, so they centrifuge it out and they get 2 things. So they have a liquid. Apparently I'm going to get worse at it as the day progresses. Okay, so after they centrifuge, they have a liquid which has phage parts in it. And then they have a pellet which has the bacterial cells. So the bacterial cells are more dense, so when you centrifuge them, they will go to the bottom. The phages are so small that they're going to stay suspended in the liquid. And what they found was when they marked the proteins with radioactive sulfur, at the end, they had the radioactive phages, the phage coats were in the liquid, and there was no radioactivity in the E. coli. When they looked at the radioactively labeled nitrogen, same thing happened. They did the same thing, but then they found the radioactive cells in the pellet, the E. coli. and no radioactivity in the liquid, the supernatant. So what that told them was when the DNA, when the phages attached to the E. coli, they're injecting their DNA into the DNA cell, into the E. coli cells. That's where all the radioactivity is that gets passed on to the cells. It's not in the protein coat. So when they radioactively labeled the protein coat, these infected cells that were infected by the virus did not produce, did not have any radioactivity in them. So these E. coli, they let them go on to reproduce and they would make more viruses. So when they made new viruses, they were not radioactive. When these E. coli cells reproduce, because that's what a virus does, it takes over and reproduces in your cells, kills the cell and makes new viruses, they produced radioactive viruses. So that meant that it was the DNA of the virus that was being passed on to the next generation. So again, just confirmation that it was DNA and not RNA, not protein that was being passed on. So at this point, we're like end of the 1940s. I did not write down a year for them, sorry. We're at the end of the 1940s. Everyone's pretty much okay with the idea that it's DNA that's the heritable information, but we still don't know that much about DNA. The structure of DNA was not understood, so it became a race at this point. A race to figure out the structure of DNA. So there are multiple groups working around the world trying to be the ones that figured out the structure of DNA. So they were using this research of Chargaff, which he doesn't really fit into, I guess he contributed to this, so we're going to put him in this one. He did experiments to figure out the composition of bases in cells. So he analyzed the DNA from a whole bunch of different species to figure out how much A, how much T, how much C, and how much G is it. And he noticed two patterns, and he gets credit, he gets two rules named after him. He found that when he looked at different species, they had different amounts of A and T and G and C. So he found that the base composition varies between species. So a tiger might not have the same composition A, T, G, and C like ratios that you would find in a mushroom. So base composition differs between species, but he found that in general, the amount of A is the same as the amount of T, and the amount of C is the same amount as G. So this is a saying, I guess, that might help you remember that I cut out. In DNA, A plus G is always equal to T plus C, kind of rhymes. A and G, T and C, kind of rhymes. But when A is equal to T and C equals to G, this is what we call the base pairing rule. Base pairing rule. So the idea that they're equal to each other means that they must be pairing with each other. So this was a big step towards determining the structure of DNA. And it came down to two main teams that were working, Two main teams that were working on it. One team was Wilkins and Franklin. Both of these teams were working in England. They were doing X-ray crystallography. So they were forming crystals of DNA and then taking X-ray pictures of them to try to figure out what the DNA actually look like. We're not at the point where we had microscopes that could see that level. Not that our microscopes are great at seeing that level still. But they were using information and data to try to figure out, and this is the most famous picture. This is photo 51. It was taken by one of Franklin's lab associates, whose name I just forgot. And it shows you that X shape that was really helpful for the other team that was working on this, who you might have heard of a little, might be a little more familiar with, which is Watson and Crick. Watson and Crick weren't doing a lot of experiments. They were taking information from other people and building models. And they were using these models to try to say, okay, we know this. So if we make the model this way, does this make sense? But no, that doesn't make sense. That doesn't work. They know the ATGC base pairing thing. And they were they're both working in England, as I said, and Wilkins, who was working with Franklin, showed that photo 51 to Watson and Crick without Franklin's permission. And that seems to be what cracks the code. So when Watson and Crick saw that picture, and based on all the other information that they had, they were the ones that are credited with figuring out the structure of DNA. And that was in 1953. So in 1953, they solved it through modeling mainly. With the help of the data that they had and with the help of the picture that Wilkins showed them. In 1962, they got the Nobel Prize for determining the structure of DNA, along with Wilkins. But Franklin did not. Anyone know why Franklin did not get the Nobel Prize? Because she was dead. Okay, you can't get a Nobel Prize posthumously at that time. You could not get a Nobel Prize posthumously. And she died from cancer, which is possibly really ovarian cancer, which is maybe related to the amount of exposure she had to X-rays through her scientific research. So she did not get the Nobel Prize, but Wilkins, who showed the picture of Watson and Crick without information, did get the Nobel Prize. There's lots of drama about it. This basically started what we call like the genetic revolution and our understanding of how cells work, how information is passed on. And the structure was, again, it was a really big deal to figure this out. And that 1962 Nobel Prize, if you do the math, it wasn't that long ago. Maybe you feel like it was a really long time ago, but we're still talking about less than 70 years ago. We figured out the structure of DNA. And everything that we're going to talk about in chapter 17 and 18, well, 16, the rest of 16, 17, and 18 is based on this knowledge that we've had for 70 years. And that's all. So let's talk about the structure of DNA. We'll get through the structure of DNA and we'll start replication a little bit. We have alluded to them before. We've talked about some about the structure of DNA. We're going to get a little bit more detailed in our discussion of the structure of DNA and our understanding of how DNA is used in the cell to make more DNA. So we'll start with the basics. DNA is a polymer. So it's a two-stranded polymer. What do we mean by polymer? It's a whole bunch of monomers stuck together. Okay, what is the monomer for DNA? It's a nucleotide. So the monomer is a nucleotide. What do you remember about the structure of nucleotides from way back when, like chapter 3 or 4? What are the 3 parts of the nucleotide? We have a house, which is the sugar, which in DNA is deoxyribose. Attached to that, we have the pool, which is that's your phosphate group. Yep. And attached to that, the driveway. That's the nitrogenous base. So that's the nitrogen base that's attached, and there's 4 bases. A, T, C, and G. Which we always represent with 4 different colors. So this coming along the sides here is what we call the sugar phosphate backbone. We've got a sugar phosphate backbone, and those bases are attached to the sugar. And they run what we call anti-parallel. So when we look at the structure of the sugars, we number the carbons. So it's a 5-carbon sugar. But one of the carbons actually comes off here. So we number the carbons starting, have you done this in chemistry? We number the carbons starting to the right of the oxygen. So like this is the 1st carbon, 2nd carbon, 3rd carbon, 4th carbon is here, and the 5th carbon is the one that's sticking out. So 1 prime, 2 prime, oops. So one of the strands runs 5 prime to 3 prime. So the 5 prime carbon, this is the 5 prime carbon that the phosphate group is attached to. And the other one at the other end, this is the 3 prime carbon, which has an OH group attached to it. So this strand runs 5 prime to 3 prime. This strand runs anti-parallel to it because it's running 3 prime to 5 prime. So they run in the opposite direction. So the orientation of the sugars is flipped on the other strand. So they're anti-parallel. That's going to be really, really important when we talk about the replication process. That's why we're making a big deal out of it. So on the inside, these are the nitrogen bases. They're hydrophobic. So they're protected from the wet water environment of the outside of the nucleus by being on the inside. What kinds of bonds are holding these together? What kinds of bonds have we talked about like a million times when we talked about water? Hydrogen bonds. So these are held together by hydrogen bonds. What do you remember about hydrogen bonds? Are they strong or are they weak? On their own, they're pretty weak, but when you put them together, they can be stronger. So you'll notice that A and T, there's 2 hydrogen bonds bonding A to T. There's 3 hydrogen bonds bonding G to C. A and G are what we call the purine bases. And T and C are the pyrimidines. So the pyrimidines only have 1 ring, and the purines have 2 rings in their structure. A and T bond and C and G bond, it's important that you know it's 1 purine and 1 pyrimidine because that's how it fits. Its DNA is not wide enough to have the 2 like T and C bond with each other. Sorry, A and G bond with each other because that would be too wide. So the width of the DNA helped, it helped Watson and Crick figure out the bonding thing and how it fit together. That the A had to pair with the T and the G had to pair with the C because of the way that the model fit together. So the strands are anti-parallel. The overall structure we call a double helix because it's got 2 backbones and they're twisted. Everything about your genes, the different alleles that you have, the different proteins and traits that your body expresses is determined by the order of the bases. So you express 1 trait and somebody else expresses another trait in your family because the A, the G, the C, and the T are in different orders in the DNA. You are not a mushroom because the order of the bases is different in your DNA than it is in the mushroom's DNA. I think it's mind blowing that it's the same four bases that determine all of that. You have in your body somewhere between. I gotta find my number to make sure I give you the right one. You have about 6 billion base pairs in your body. So 6 billion of those A's, T's, and C's and G's in your body. And E. coli has about 4.6 million. So you have about 1,000 times more DNA than a bacterial cell, an E. coli bacterial cell at least. So just when you thought we were done with the history part, we're not. So once I figured out the structure of DNA, knowing the structure of DNA was really helpful in figuring out how it worked and actually how replication worked. Watson and Crick did a lot of work, again, theoretically thinking about how can we take this to make more of this in an accurate way. So mistakes sometimes happen in DNA replication. In your body, it's about 1 in every 10 billion base pairs if there's a mistake. Okay, I don't know, I can't imagine doing something 10 billion times and only making one mistake. But your cells do that all the time. 1 in every 10 billion base pairs makes a mistake, and often your cells can correct that mistake too. So the structure of DNA was helpful in figuring out how it was able to replicate. But before we knew how it was able to replicate perfectly, before we understood it as we do, Messelson and Stahl did their experiment, and they were determining how does DNA replication happen, and there were 3 options. One was could be conservative, or it could be what we call dispersive, or it could be semi-conservative. So they're the ones that figured out which of these 3 options it was. So if you look at conservative, the idea of conservative replication would be you take one DNA molecule intact and you still have it intact as you keep replicating, but you just somehow make a copy of it. But you're keeping this original molecule intact and just making a copy. So there's the parent molecule stays intact. It stays together the whole time. So it's somehow taking this molecule and your cell looks at it and makes a new one without changing this in any way. Another idea was that maybe it's dispersive, meaning that as DNA replicates, you take pieces. So each new molecule has pieces of old DNA and pieces of new DNA in it, but not necessarily in any kind of, not that it's not organized, but it's not like, it's just like chunks. You take chunks and you put new chunks in, and then you end up with a mixture of old and new in the new DNA. The third idea was that it was semi-conservative, that this original molecule was taken apart and a new strand was made from it. So this was that the parent strand served as a template to make the new ones. So each new molecule is half old and half new. And they did this the same way or similar way to what Hershey and Chase did. They labeled the DNA with heavy nitrogen, which is radioactive, and then they watched what happened and looked at the offspring, the daughter DNAs, and they found out that they were a mixture of red and blue. If we're saying that blue is like heavy nitrogen and red is light nitrogen, they ended up with this mixture of like normal nitrogen and heavy nitrogen, so that they ended up determining that it is a semi-conservative replication process. So that means that each strand is a template for the new DNA to be made. So this was the winner. So DNA is a double helix with strands that run anti-parallel that replicates semi-conservatively. So this is just giving you a real quick overview of it from a prokaryotic and a eukaryotic standpoint. So this is prokaryotic. So DNA replication has to have a starting point. So when we look at DNA of a bacterial cell, so a prokaryotic cell, the bacterial chromosome is a circular shape. Our chromosomes as eukaryotes are linear. But again, this idea is the same, that you have a template strand. Your replication I lost the word, replication complex. Replication, there's a word that I can't come up with right now. Gonna bother me. Mechanisms. The replication machinery is going to bond at this origin. In terms of prokaryotic cells, they only have one origin. For eukaryotic cells, there's multiple origins. So there's multiple places where this machinery can bond and start the replication process. So in this case, we have the outer strand is the parent template. And this inner strand that you can see is a lighter blue is the daughter strand. So the replication machinery is going to start here, and then it's going to work in both directions. So here where you see this starting to split, these are called the replication forks. So the replication machinery is going to come in, it's going to break those hydrogen bonds and start unwinding the DNA so that you get these this is called the replication bubble. And it's just going to copy the chromosomes so that you end up with a new strand and a template strand, the parent strand and the new strand. And you end up with two identical chromosomes. So neither of them is fully new. You've got half old DNA, half new DNA. Half old DNA, half new DNA. Because it's semi-conservative. Same thing's happening here, except it's happening linearly. It binds at the origin of replication. You've got the old strand on the outside, the template strand, the parent strand, and the inside is going to go in both directions until they meet in the middle, and you're going to end up with these new strands of DNA, which are parent and daughter, so semi-conservatively replicated. So we're going to talk about the specifics of how that happens. We're going to get through this slide probably only. Okay, there are 7 proteins or enzymes that are involved in this process. So you're going to keep track of them. How many are there? 7 enzymes or proteins. All of but one of them are enzymes, but. So this is showing you the structure of DNA, and this is the beginning process. So DNA is normally wound up. So what's the first thing that's going to have to happen? How does this look different than that? It's going to unwind. So it's going to go from being a twisted ladder to being a ladder. So there's several actually enzymes that are involved in that. It doesn't want to untwist. So the first one that we'll talk about is DNA helicase. Helicase is responsible for unwinding DNA. At that replication fork. It's also going to break the hydrogen bonds between the bases. So this is our replication fork. And the DNA helicase is going to untwist it, and then it's going to start breaking the bonds between the bases. And the blue and the green ones come apart better than the red and the yellow ones. One in 10 billion is how many mistakes your cell makes. I can't even get 5 done without making a mistake. I broke it again. Oh, I did it again. So the DNA helicase is in there, unwinding and breaking the hydrogen bonds between the bases. What do you notice about the end here? It doesn't want to stay unwound. Okay, when you are unwinding, it's creating tension, and that tension actually has to be relieved by another enzyme, which is topoisomerase. So it keeps it from retwisting on itself. So helicase is one of our proteins, our enzymes. Topoisomerase is the second one. So as this is unwinding here, this is keeping it from rewinding. It's preventing that from rewinding. The other thing that's important here is these little binding proteins. They're there to just keep those hydrogen bonds from reforming. Okay, so if they got too close together again, they might form another hydrogen bonds. So they're going to keep the strands separate. So they're going to be on every single base as it unwinds. So we've unwound it. We've broken the hydrogen bonds. We're keeping it from going back together. To really get the process started, we need our fourth one, which is the primase enzyme. We're ready to start replication, but replication can't just start on a single strand. It has to start where there is a double strand. And this I think is weird, but the primase is actually going to bind RNA bases. See how it's bringing in these red ones? It's bringing in 5 to 10 nucleotides of RNA that are going to match with these bases of the DNA. And that's going to be the primer. That's where all the replication machinery is going to start assembling to do the replication process. So they are RNA primers, they're RNA bases, 5 to 10 nucleotides long. They're complementary, meaning they still match up in terms of A and C and G and T, except that there's a U in RNA, which we'll talk about a little bit more next chapter. And the reason that you use the RNA is more available in the cell, is what I found when I started researching that, because I said that's weird. Why are we not using DNA? But it's just that it's more readily available in the cell. That is where we will stop today. On Monday, we're going to dive in and really get into the details of replication, and your homework will be due.